I don't know, and I don't know why it just kind of is a fluke when it happens. So is what it is. Okay, so we are doing nervous system today, and nervous system is the toughest lab overall for me. You may think musculoskeletal is for you, but it's very likely you're going to find that this one's a really tough one too. We have not been through it in lecture, but we will start it on Monday. So before you have lab next week and before you have the quiz, you will have an opportunity to go through a lot of this again. But this is, just like the nervous system, is challenging. Okay, so there's a lot of parts and pieces to it. So I had an entire lab last semester that was all online. So I left all the parts and pieces there. Okay, so we've been in there before, you know what that looks like. So there's videos and all that kind of stuff. But this time I had someone, I'm really lucky because the stockroom person, one of them, lives in Clayton. So she brings me stuff if I need it. And so this time I asked her to bring me some special stuff. I have models of the brain, an eye and an ear that I'm going to go through. I also have sheep brains, and I have some eyes. I think these are sheep eyes, but I'm not quite sure. Oh, I totally forgot. I'm glad Rose is back before we start. You back, Rose? Whoop, where'd she go? All right. So anyway, we're going to start, the lab starts on page 143. And I have a couple, so I have 3 different cameras, and I have my entire kitchen counter full of stuff. And I didn't have room for everything, so I have some over on the side counter. So you're going to have to bear with me a little bit. We have, in the lab, we have some activities we can do. A couple of them we'll be able to do here, but most of them are activities that we won't be able to do. If you read through them, you probably could do them at home if you're so inspired. But otherwise, we'll do a couple activities together. Otherwise, you're stuck listening to me. Lucky you. So next week, recognizing this is a really tough topic and quiz, I'm not going to give you a word bank, but I will let you use your resources. Okay, time is still going to be an issue, but that will allow you to go directly to the word bank and to look stuff up in the chapter. You could use your text, but as I say, time will probably be an issue for too much of that. Okay, but next week will be open book, open notes. So keep that in mind as we go through this. And we're going to start by talking about the brain. But before we talk about the brain, let's put the nervous system in perspective. We have, I've talked just kind of here and there about the nervous system, but the nervous system includes the brain and spinal cord, but then it includes all sorts of neurons that run all over the body. And you've also heard the term nerves. A neuron is an individual cell. You know that nerves are usually groups of those neurons that run together. So we've talked about sensors when we've talked about the skin, but there's sensors all over the body. So when we start talking about the nervous system, we're talking about all of that. And we divide it in 2 parts. The first one is the central nervous system, and the central nervous system is exclusively the brain and the spinal cord. That's all that's in the central nervous system. The peripheral nervous system is everything else. Okay, so we're going to start by looking at the central nervous system, and we're going to start with my brain model here. And let me switch and see what picture I like best. I think it was this one. Nope, it wasn't. Got to figure out which of my 3 works best. Okay, so this is a brain model. And so if this would be the front, this is the back of the brain. Underneath here, this is called the brain stem, but that would be headed down through the foramen magnum and into the vertebral column, right? That would be what constitutes the spinal cord. So with this model, you're now looking at the front of it, and the names of the lobes are really easy on this. Okay, and if you want to follow along, page 147 has a picture of a model, okay? Versus, we'll look at the sheep model in a minute. But this is the front, and this is called the frontal lobe. Okay, just like the skull had the frontal bone, under the frontal bone is the frontal lobe. And the frontal lobe is separated from the parietal lobe by what's called the central sulcus. And all of this is, you can tell a little bit here, but it's sort of like mountains and valleys. And the sulcus is the valley. So there's gyrus, which are the mountains, and then the sulcus, which are the valleys. And all of this is going to be white matter. And it doesn't matter. They don't show it on here, so we won't talk about that. But this entire area that you can see is called the cerebrum. And it's made up of these gyrus and sulcus. And that increases surface area. But there are 2 halves of the brain. You can actually, now this is a model, so obviously you can do that, but both halves are identical. So we can change to looking at this half, and the other side is going to be identical. Oh, I forgot though. Wait. I got partway through the lobes. Okay, so we have the frontal lobe. We have the parietal lobes, right? And then we have, guess what the back one is called? Guess what this one's called?
Occipital.
Occipital, just like the bone. Now there's also one more part on the side. Guess what that one's called? Come on, Kelly, you're on a roll.
Temporal.
Temporal, right. So those are easy. You should know those if you know the skull, right? So if you remember the skull bone. So those are the different lobes. There is one more lobe that we need to know, and it's this funny looking thing back here. Okay, it's on both sides. That is called the cerebellum. So when I said this entire portion is the cerebrum, that's cerebrum. This part in the back, that's a darker color here, is the cerebellum, bellum, okay? And those are lobes as well. They're just a little bit different, right? So now let me take it apart and let's look at the cerebellum. The cerebellum has this really funny shape to it. It looks kind of like, anybody heard of an arborvitae? A plant called an arborvitae? That's actually, they call these arborvitae, and that's a characteristic morphology that's seen in the cerebellum. This is the brainstem, and the brainstem has 3 parts. It has a midbrain up, kind of in the midbrain, and then it has this little kind of funny shaped bulge that's called the pons, and then down a little bit lower, there's another little bulge. and that's called the medulla oblongata. Okay, sometimes that's shortened to medulla. And when we look in here, there is a lighter part that I'm going around. That is called the, oh, now I'm gonna have a total brain cramp. It's a, wait. I'll come back to it. But inside this is the lateral ventricle. And the lateral ventricle, there's actually four, I think there are 4 ventricles. And the ventricles are openings. And they are responsible for making spinal fluid and bathing the brain and the cells in spinal fluid. So that is what you're seeing here. And there's some other, you'll see other spaces that will tell you like they're the 2nd, 3rd, and 4th ventricle. This thing that is hanging off here that looks kind of like a kidney bean, that's the pituitary. And the pituitary actually does hang kind of off the brain down behind the nose. Above the pituitary is the hypothalamus. That should sound familiar. Does anyone remember in lecture what we credited the hypothalamus with? Homeostasis. When we talked about temperature, we talked about the hypothalamus being the control mechanism in the brain for homeostasis. That's its claim to fame. Above it is the thalamus, and the hypothalamus and the thalamus are actually areas of the brain that are called the diencephalon. And the thalamus is in an area where it's going to control everything coming up from the brainstem. So all the activity coming up from the brainstem going into the brain is going to have to relay through the thalamus. Okay, so it's kind of considered the relay station. So, all right, I got to look it up, and I'm going to be, where are ya? Oh, the corpus callosum. I kept wanting to call it corpus luteum. The corpus callosum is this, and this one piece goes right through to both hemispheres. So even though you could separate the brain into 2 parts, you would get to the corpus callosum, and that's responsible for communication between both sides. Okay, so those are the basic parts of the brain. I'm going to switch, and we're going to identify these same parts on the sheep brain, because this is real brain tissue. Give me a second. I've got all my PPE on. At least I'm working on it, I should say. These smell because they're in preservative. They're not in formalin. They're in a different type of alcohol-based preservative. But my son came down to have lunch, and these were out down here, and he thought that was less than exciting in the kitchen. I did invite him to look them over and help, but he decided not to. But these keep it wet. They're soaked in preservative so that they don't get dried out. All right, so this is a sheep brain. You're looking at a sheep brain. So basic parts of the sheep brain are very similar to ours, but you may notice that you can't see the gyrus and sulcus very well. You can see them down in there, but this brain is covered with meninges, and meninges are the covering that protects the brain. So if you were to look at basically from your hair down to the brain in the skull, if you made a hole and you look down, what you're going to see is you're going to see let's say here you would see the parietal bone, right? You'd go through the parietal bone. The next thing you would see is the meninges, and it's 3 layers. So as far as humans go, if you've heard of something like meningitis, that's inflammation of the layers of the meninges. That's an infection in those layers. So this is actually covered by the meninges, and it's a very, very tough layer. I can't even, let me see if I can find a little piece. It's very tough. I can't rip it, okay? And you really like to be very happy about that it's such a tough layer. If somebody gets hit on the head, for example, you know, sometimes you'll hear they get a bleed in their brain. Sometimes that bleed is actually within the layers of the meninges. There's a place called the arachnoid space, and that space, if blood gets in it, there's no place for it to move as far as if you think about the skull, right? But it can depress the brain tissue. That's why those are so serious. All right, so orientation here, we're looking at the front, so the frontal lobes. The distribution of brain tissue is different in a sheep than ours. Most of the frontal lobe for us is processing and thinking. Those are higher functions, language. The sheep lacks that, so they have a much smaller frontal lobe overall than we do. But they'll have a parietal lobe, and there's two of them, right? One on each side. And they'll have a temporal lobe as well. And as we move back, they will have an occipital lobe, and they will also have a cerebellum. And I'll be able to show you that better on the half brain. But also on here, underneath this lump, you see that little lump there right in the middle? That's the pituitary. Okay, and ours is a little like that. Okay, it kind of hangs down off the brain. This is held by the meninges, so it doesn't dangle particularly. And then here, the lobes are a little big. Let me see if I can, maybe I'll point with this so that I can do this a little more easily. So here's the pituitary. Here is a nerve that's coming in. And this is where it comes together. There's actually two nerves, one coming from this way and one coming from this way. Any idea where those might be coming from? You picture where, let's see, there's the orientation of our sheep. That's looking at you.
The eyes.
Where?
The eyes.
The eyes, right. This is the optic chiasm. And this is where the nerves from each of the eye come together. And then this doesn't show as well as I would like. One of them doesn't show at all, but there is a flap right here, okay? I'm not sure how well you can see it. It's just a little flap. And that actually is responsible for smell. It's called the olfactory bulb, okay? And most animals have better smell than we do. So if you look at a dog's olfactory bulb, for example, it is, or its lobe, it's going to be much bigger than ours. So there are, let's see, I don't think I want to, I think we're all set with that. Let's see if I can, not really well. Okay. And then when we look at this, now it's cut down the center. There is that arborvitae. Okay, so that's the cerebellum. Cerebellum is for us things like balance and smooth movements that allows us to move smoothly. So there's the cerebellum. And you can see the difference in the amount of tissue there is, right, in the cerebrum. But here is the corpus callosum, and you can actually see there is a band of tissue right there that runs along. And then there's this opening. And this opening is where the lateral ventricle is. Okay, so that is an opening, and we would expect spinal fluid to be made there and to run through there. As we move down a little bit, parts that we can see well, this is a little mushed, but if you get way down here, that's part of the spinal cord. But there's a slightly, I don't know, widened section right here. That's the medulla oblongata. The medulla oblongata is responsible for internal organs and function. So we'll talk about that in a minute. But then we move up toward the brain, and there is a widened section here. That's called the pons. And then we move up into what's called the midbrain up here. And it's a little harder to see on here, but there is a thalamus and a hypothalamus, but they're much smaller. I wouldn't be able to identify really where they are. This looks like it's probably the thalamus, which would make the hypothalamus below it, but I can't see well enough. Okay, so there is our sheep brain. Trying to think if there's anything else I want to go over on that, but I think that's good. And while we're here, I haven't decided, this is an eye. Okay, so there really is only a couple things without opening the eye, and I do have a scalpel if we decide to open the eye and look inside. But if you think about your eye, you have the white of your eye, right? So if you open your eyes in shock, you see the whites of your eye. The white is a protective layer called the sclera, and that's all here. Okay, this has been preserved once it's been removed, and the fact it's been preserved makes it look a little wrinkly. It also has the cornea. The cornea is the outer portion on your eye, and that's protective as well. Underneath the cornea, there would be a compartment, and that compartment has a little bit of jelly-like fluid in it. And that's called the aqueous humor. But then if you look at your own eye, I'm sure you've done it in the mirror before, you have an iris. And the iris is the colored part of your eye. When someone says, what color are your eyes? If you've got blue eyes, it's because your iris is blue. But you have something in the center of your iris. Anybody know what's in the center of your iris? Your pupil, right? Your pupil. And that's controlling the amount of light that's going into your eye. On the back of this, there's a piece of tissue right here. So here's and honestly, I don't know, there's no eyelashes, so I can't tell which direction it goes. But here's my cornea. So it's right on the back, there's this right here. And that actually is the nerve that comes out of the eye. So we saw where that went into the brain, right, at the optic chiasm. So that's a nerve that comes out. There's sensory receptors in the eye, and the sensory receptors are what is going to be triggered and send the message to the brain of what we're looking at. So we'll go through a model of that. Anybody want to see what's inside or not? You do? All right. We'll see how I do. That's why I've got my goggles and everything on, because this, I think, may squirt. Wondering whether to go directly. I think I'll go from the back, or I'll go from the top, and we'll see what it does. If I scream, oh, it didn't squirt, but there is fluid in it. Oh, it does squirt a little bit. I take it back. So let me make this a little bit bigger cut so we can see what's in here. If you move to, I can't turn my pages right now because my hands are all yucky, but if you move farther along by a couple pages, you will find a model of the eye, and maybe you'll be able to see what we're actually looking at. I guess I'm going to have to be a little more. Okay, all sorts of fluid in there, but let's see what we can see. All right. So from this side, oh, this is cool. Okay, so this is basically right down the center. And what I'm looking at, a bunch of fluid came out. There's a whole bunch of jelly-like fluid that's in your eyeball. Okay, so right behind the cornea, there's a little, probably not in here, I'm guessing, but normally in a healthy eye, you would have a little bit of jelly-like fluid between your eye and the iris. Let's see if I can. All right, well, that's not showing me much, but there would be a little bit of fluid there. Then you would get to the iris, and the pupil's a hole. And behind the pupil is the lens of your eye. So that would, the lens would be behind the pupil. And you're actually looking at the lens right here. This right here is the lens. Okay, so it's kind of, these are opaque probably because of the storage, but there's the lens. And the lens is what changes your vision. And so if you're, you actually, when you're, well, we'll talk about that in a minute, never mind. And then in the back of your eye, there's all of this, it's kind of a different color, but that's the retina. And the retina has the cones and the rods. And the cones and the rods are the sensors for the eye. The cones are going to sense color, and the rods are going to see in black and white. But those two are both going to be triggered in the retina, and the message is going to be sent out in the back through the optic nerve. Now, what I do want to see, oh, and we can see it here. This is awesome. I don't know that you can see it. Let me see if we can. There is a spot right here. Let's see if I can get it just right. And it's a little tiny spot in the retina. And that's where the optic nerve comes in. So although we wouldn't notice it, we actually have a blind spot there where the nerve comes in, because light, there's no cones and rods to pick up any vision at that point. So we actually have a blind spot, and we'll talk about that, but we can actually see it in there. All right. So anything you want to see before I take off my gloves? Want another close-up of an eyeball or brain parts? Okay. Now let me move that. And just a sec, I'm going to wash my hands. Okay. We'll get rid of the glasses. And let's go back to, I know you can't see this very well, but I'm going to put it there for just a minute as we go through. I want to make sure that we talk about some of this, not just look at it. Wonder, hang on. Let me see if We'll see if we can do that. And at least you can see what page I'm on, okay? I don't think you can read it. But as we go through 144 and on page 144, things that I want you to know the function of, and you'll get some practice in a couple pages, but we talked about the ventricles, so put a little star next to the ventricles. We know that those are where we get cerebrospinal fluid. But then go down underneath cerebrum, and we talked about these 4 lobes. So you do not need to know everything about these. But what I do want you to do, I'm going to give you some stuff. If you have a highlighter or you want to run and get a highlighter, I'll give you certain things to highlight. And when we do that, that will allow, I will ask you those specific things. I won't ask you, there's a whole long list of stuff I won't ask you. So see if I can, I don't think you can see that either, but so under frontal lobe, controls motor functions and abilities to think, problem solve. Okay, controls motor functions, abilities to think, problem solve. On the parietal lobe, receives information from sensory receptors located in the skin and the taste receptors. And then the occipital lobe interprets visual input. And on the temporal lobe, sensory areas for hearing and smelling. And then down below, we have the diencephalon, which I already mentioned was the thalamus and hypothalamus. And under thalamus, find receive sensory impulses. And then at the bottom, it says gatekeeper to the cerebrum. So receive sensory impulses, gatekeeper to the cerebrum. The hypothalamus controls centers for appetite, body temperature, water balance, and its primary function is homeostasis. The last sentence of the cerebellum coordinates equilibrium and motor activity to produce smooth movements. Okay, any questions about those?
What was the hypothalamus again?
The hypothalamus is control centers for appetite, body temperature, and water balance. Its primary function is homeostasis. So if I ask you anything about what they do, it will be from here, okay? Because these are things we don't really cover in class. The last one is the brainstem on page 146. And under brainstem, well, let me see if it's, hold on. Now, I guess we got to do under brainstem. Centers for functioning of internal organs. And relay station for nerve impulses passing from cord to the brain. That's the general function of the brainstem. And then the midbrain relay station. And the pons is passageway for nerve impulses. And C, the medulla, controls internal organs. For example, cardiac and breathing centers. Okay, the rest of these 2 pages you can do on your own, and they'll be good practice to prepare for the quiz, because they do talk about the different functions as a summary. And they also give you an opportunity to label the different parts of the brain. All right. Now, actually, while we're back here on the brain stem, how many of you are Walking Dead fans? Anybody?
I just started watching it. I'm on season 2.
Then you've gone far enough. You're going to be our expert here, because in The Walking Dead, anybody, I gave up on it. I had like two times that I was like, oh my God, this is awful. And so I gave up on it. And then I'd ask everybody in my family what was happening. It was easier that way. But I watched through season 1, and they go to the CDC. Anybody remember that? So they show up at the CDC. And do you remember the doctor at the CDC explaining what was happening to the zombies?
Yeah, and he showed it on the screen.
He did. He showed it on a screen. It was a video. And what happened, Maria? What was he saying about it? Do you remember?
He said that only one part of the brain was lighting up, like most of the brain was dead still. And then there was like one part, which was like the very middle, that was lighting up, and that was coming back, I guess. That was working the whole body.
Right.
Yeah.
Now, this, of course, is a show, but what they did was you saw the person die and you saw the whole brain go dark because there was no brain activity. And then after a little bit of time, you see a little light starting to come back on, showing brain activity. But it was in the very center. And where it was what they were trying to show was the brain stem. And the brain stem controls those very basic things. You'll hear sometimes that someone is brain dead, for example. And what that means is the functioning of their cerebrum is severely compromised. But frequently, at least for a time, their heart and their lungs can still function. That's because that is a very primitive function of the brainstem, is to protect those internal organs. And they are so far in and underneath that they're protected really well. And that's where the centers for breathing and for cardiac activity are. So what they were showing was these zombies are coming back, but they don't have anything that allows them to think. They don't have anything that is high functioning. The only thing that they have is that very basic minimal activity. Now, you wouldn't be walking around and you probably wouldn't have appetite, but still, that's the idea, is that that cardiac activity and the breathing comes back, but the personality and who that person is not there. So anyway, there you have my lesson for today about the brain stem. So let's move on, and I want to talk about, I have to tell you, I ordered a document camera. I am about as technologically savvy as nothing. So I'm reading all this stuff and going, I have no idea what that meant. So I haven't gotten it yet, and I really could use it today. And of course, everybody wants a document camera is the problem. So I have all sorts of handy dandy little things today to help. I have a little whiteboard. And I want to talk about reflexes. We're getting into the peripheral nervous system. We've talked about the central nervous system. We're not going to really be talking about the spinal cord specifically. Let's see, okay. But on page, well, I lied. We are going to talk about the spinal cord. I'll do that before I do reflexes. That's important. So there's a picture on page 148. And the spinal cord kind of looks like, not really like that, but it's also got kind of a butterfly shape within it. Okay, in the center. And this is gray matter. And then this is white matter. And the reason that they're different colors is because white matter has myelin. Remember we talked about myelin in class? And we talked, it was in lab, I think, when we talked about neurons. Our neuron cell has a myelin sheath. And when that myelin sheath is compromised, that's when people have multiple sclerosis. Okay, so all of this area that's got the white is covered with myelin. But these center part don't have myelin. And we're going to look at a reflex. Okay, and a reflex, first of all, if we look at, here's the spinal cord, we actually have, and I didn't cover this, I wonder if I probably missed it a couple pages ago. Yeah, they've talked about there's 12 sets of cranial nerves, and there's 31 sets of spinal nerves. Those are nerves that are going to come off the spinal cord at different locations between the vertebrae. So let's say you step on a pin. You're walking around the house and you're barefooted or stocking footed and you step on, or a Lego, that's even better. You step on a Lego. Those are god-awful things. So you step on a Lego on your foot. Normally what then happens is the message, so this is my Lego. I don't know if you can see my Lego very well. But here's my Lego down here. And that, whatever pain sensor, right? That's probably what it's going to be in my foot. That pain sensor is a nerve or a neuron, and it is going to go all the way up my leg. And it is going to go actually into this gray matter. And in that gray matter, a couple things are going to happen. First of all, if this, I didn't step on the Lego hard, so I'm not jumping off of it or anything. I just go, oh, something's under my foot. This message is going to get in the gray matter and it's going to go all the way to the brain. And that's not going to be a pain sensor. That may be a pressure sensor. I know I've stepped on something and I'm looking at it. So it's going to go all the way up my spinal cord to my brain. And my brain's going to process that. And the message then, my brain is going to say, oh, you stepped on something. Let's look and see what that is. So that message is coming back down here. And you want to move your leg now. So the message comes all the way back down to your leg. So you lift up your leg to look. Okay. So this particular path where you felt the Lego is called a sensory neuron. Okay, and that sensory neuron takes the message up to the spinal cord and to the brain. This other path that brings the message down from the brain through the spinal cord and all the way to the quad, right? If I want to lift up my leg, my quadriceps, so I can lift up my leg, that is going to be a motor neuron. Okay, so sensory brings the message to the brain, motor sends the action out. Okay, so now that's the basic system and how it works. But there is something else here, and I'm going to leave this. Actually, I'm going to take this out for now. And let's say, first of all, you've all been to the doctor, and there's a video on this in the lab, but you've all been to the doctor and he whacks your knee with a little hammer, right? Anybody not had their knee whacked with a little hammer? They may actually do one for your arm too. But when they whack your knee with the hammer, you're not thinking about it, right? But your leg kind of jumps. Okay, your leg extends. That is called a reflex, and that is what the doctor's looking for. You have a number of different types of reflexes. So let's say now we're at your patellar tendon, because that's where it's being done. And you know where the patella is. And the tendon is just below the patella, goes down to the tibia. So here is our patellar tendon, and you hit it just right with that little hammer, and the message is going to go up to the spinal cord. Okay, that's normal. That happens no matter what the sensor is. But a reflex is something that happens automatically. So in this case, and I'm just going to change the way this looks a little bit. This message is going to come up into the spinal cord, and there's something called an interneuron, I-N-T-E-R. And an interneuron is a neuron that is entirely in the spinal cord. Okay, so it starts and ends in the spinal cord. And what happens is this sensory neuron triggers this interneuron in the spinal cord, and the message is sent directly back, and your knee jumps. Now, what you probably can recall is your knee jumps, and then in your brain you say, oh, he hit my knee and my leg moved. You weren't thinking about that. Your brain didn't get that message until after your leg moved. So the message is still going to go to the brain, but instead of the brain being what controls your knee jumping, there's actually right in your spinal cord that's controlled. So when you have a reflex of some sort, so when your doctor is checking for reflexes, he's checking for how this path is working. Okay, so that is a reflex compared to the usual way a message comes in, and that is that the sensory neuron will bring it into the spinal cord. It will go to the brain first. The brain will send down a message, and you'll move, okay, or do whatever you need to do to accommodate that message. Okay, so that's the difference between how a normal message is sent and how a reflex occurs. All right, so I think that's all I really want to do on that. Are there any questions about the brain or the spinal cord or the path of nerves and nerve impulses? So you'd be able to tell me a sensory versus a motor action. Okay, so we'll talk more about that next week anyway. Let me condense that. And let's move on to the eye. We already had the cow eye or sheep eye. I'm not sure which it was. I think it's a little small to be a sheep eye or a cow eye, sorry. But let me try this video. Let's see if this one works a little better. Oh, all right. So here we're looking in the eye. Okay, so you're seeing the sclera or the white of the eye. Now there's a lot of parts I'm not going to cover on here, but if you notice at the end of the chapter, you have all of that. You have what's expected of you that you'll know. Okay, so as I look at this, and the sclera is the white of the eye, I'm looking at the cornea, which is that outer protective layer. And if I turn this all the way around, that is where the optic nerve will come out of the eye. I'm going to turn back this way and take this apart. And so we're looking right now at the upper part, and this is the retina. Okay, this is where all the sensory, the rods and cones are. So those are the senses in the eye for sight. You can see that's where the optic nerve comes out. And then I'm going to take out the cornea. And this is the iris. That's what gives the color to your eye. This one is brown. You can't see it very well in this light. But notice there's the pupil in the center. And you'll hear on, if you watch any sort of show, they'll say the pupils are dilated. That means that the pupil gets larger. You know that happens when you go out at night, for example. Your pupil gets larger because it's trying to take in more light in a low light environment. Then behind that is the lens, and we got to see that on the sheet. Okay, and notice that the lens, whereas this is curved to match the shape of the eye, this is a lens. Okay, and it's convex. I had to think about that for a second. That's convex, and that's going to be what determines how well you see. And then in here, this is just meant to show you that there's fluid in here. So the rest of the eye, this interior, is going to have this fluid. Now I didn't show you, let me go back very quickly and show you that between the iris and the cornea, there's space there. And that's got aqueous humor. Aqueous is where we hit first. This is vitreous humor. Both of them are jelly-like and just a viscous fluid. And then, of course, we see, just like the top, this is retina. And then we've got the other half of where the optic nerve exits. So if we were to look, and we were able to see, which I doubt we'll be able to actually do, but maybe you can see, yep, you can see through there. There's a point at which there is no cones and rods. Okay, that's the point at which you have a blind spot. So let's take a look on page 151. There is an exercise, and we're all going to try this. I'm going to read you or tell you the directions. And one thing is, you have to go very slowly. And the reason is, if you look around, you don't see a blind spot. Do you have a blind spot anywhere? I don't see any blind spot. Your eyes accommodate for that area of where the optic nerve exits the retina, or exits the eye, and where there's no sensors. So your brain is automatically compensating all the time for that. So in order to be able to see your blind spot, you've got to go very slowly. And what you're going to do, we'll just do it for one eye, and then if you're dying to do it again, you are welcome to. But what you want to do is you want to start out holding your book probably about arm's length. And you can wear glasses for this, but you want to hold it, the cross, the plus sign, in front of your left eye. And you want to close your right eye and just stare at the cross. And then you want to very slowly move the book toward you. And at some point, the circle on the left will disappear. Okay, and you have to go really slowly or you'll miss it. So go ahead and give that a shot and see how well you do. I had to practice it a few times. I did find about where it happens anyway. It only happens for just a second too, if you're moving it, so. For me, it's about right there. I don't know about you guys. I have terrible eyesight though too, so who knows? But if you're having trouble finding it, but it is kind of interesting. So anyway, you can continue to try that if you want. We're going to try another one, and with that is on page 152, and you need a pencil or a pen or something that's got a point. This one, my arms are not long enough for. It's because I'm old. Okay, so in this one, you hold the pencil upright at arm's length. And you want to slowly bring it close to you. Okay? And you want to stop as soon as it's blurry. And this goes along with age. This is one of those definitely with age ones. If your eyes are young, you can usually bring it right up close and your eyes will still keep that in focus. As you get older, your eye muscles, the muscles that are going to move the lens to a different shape to accommodate keeping this in focus, they no longer work well. Thus, you see older people, and this happens to everybody. You get to about, I don't know, somewhere between 40 and 50, and your eyes don't accommodate anymore. And so what do you see people that age have? What do people have when they get somewhere 40 or older? Happens to everybody.
The bifocals.
Either bifocals or
Reading glasses.
reading glasses, exactly. Cheaters, right? The cheaters, so that you can't see things. Your eyes don't accommodate as well anymore. So you have to have something to help. Otherwise, you're doing this, right? So you guys are all young, so that hasn't happened to you, but it will. That's why I say my arm isn't long enough for this, because as I look at it, I really can. It's arm's length, I can see it, but not real well. But I also have bifocals. So anyway, that is related to age, and it's related to the muscle. There's something called the ciliary body, and that's going to change the lens shape so that you can change your focus. It doesn't work well for older people. All right. So next is the ear. And let me put my eye back together here. This preservative kind of stinks. I'm used to it, but I'm sure my husband and my son are going to think this is wonderful when they come in the kitchen tonight. But, oh well. That's what happens when you live with a science person. Okay, so you are seeing here the inside of an ear. And on page 153, it's there as well. And this is the outside of the ear. Okay, here's your earlobe, sort of. And this is called the pinna. This is called the pinna. And the pinna is actually shaped the way it is, not to be really cool or anything, but because that actually funnels sound down into your ear. This here is the auditory canal. Okay, that's where you get earwax, right? So there's the pinna, and that's funneling sound into the auditory canal. Then it gets to the tympanic membrane. And this actually isn't the right tympanic membrane for this model, but it will do. And the tympanic membrane, that's the one, that's why they say don't stick things down in your ear, like Q-tips and stuff, because it can damage the tympanic membrane. The idea with the tympanic membrane is sound's going to come down through here, and it's going to hit the tympanic membrane. And the sound waves are going to trigger these three bones. They're called the ossicles. And they're not three bones on here. You can't see it very well. But this first bone, let me see, the first bone, let me get something easier to, is here. And that's called the malleus. And then there's the incus. And the last one is this little bone, and it's the stapes. Okay, so the malleus and incus and stapes are the three bones that are behind your membrane. And they actually, the idea with those is they accentuate the sound. They start vibrating, and that increases the sound waves. And you can't see it, but the stapes, this right here, that's the last bone. And that last bone is going to hit something called the oval window. And the oval window is on the vestibule of this structure. This structure is responsible for your balance and for your hearing. So notice this swirly end. That is the cochlea. And the cochlea looks like a little snail. And that's where the sensors are for hearing is in the cochlea. Okay, there are hair cells in there for hearing. And then this part, let me get here. This part is called the semicircular canal. And those semicircular canals are for balance. So if you have problems with vertigo, for example, that would be one of the places that they would suspect there might be a problem. So there's fluid that runs through here, and that fluid helps us. So if we know that if we lie down, our brain knows we're lying down. If we tip our head to the side, our brain gets the message that our head is tipped to the side. Vertigo is where that message is not getting through correctly, and people feel they lose their balance. They always feel like they're, even if they're standing up, they don't feel like they're standing up. They feel like they're in an awkward position. It can be awful, I understand. I hope I never have to know. So anyway, our cochlea, our semicircular canals, and then off the back, this is our auditory nerve. And our auditory nerve actually is a combination, I should mention, this is our oval window. This little spot is where the stapes hits, that's the oval window. But where it hits here and between the cochlea and the semicircular canals is called the vestibule. Okay, so there's the vestibule, and so there is a nerve that comes off the vestibule that's called the vestibular nerve, and then the other one comes off the cochlea. Now, the two of them, once they get together, are called the auditory nerve. Okay, but your book has them, I think, individually, doesn't it? Yep. Okay. Now, one other part on this, there we go. Okay, one other part on this that you should know is this piece right here. That's called the eustachian tube or the auditory tube. The eustachian tubes are those things that get plugged up when you have a cold. They may get plugged up when you're on an airplane, for example. They make or you go up a mountain and your ears pop. That's thanks to the eustachian tube. That goes into the back of the throat, and that equalizes pressure between the middle ear, which is the point behind, the middle ear is behind the tympanic membrane. Okay, so I don't know about you. Any of you have ear infections when you were a kid? Okay, ear infections are when the area behind the tympanic membrane gets fluid in it, and oftentimes that's because this area gets swelled. There's a couple different ways it can happen. It can be the grade. So instead of having a steep enough grade from the middle ear down into the back of the throat, it's not enough for gravity to help move anything, okay? Or it can be that this gets inflamed, maybe through allergies or something, and it doesn't drain. Fluid gets trapped behind the eardrum, the tympanic membrane, and that can even lead to hearing loss if it's there for a long time. And that you can see why, because you're anticipating getting that amplification of sound from the tympanic membrane through the ossicles and into the oval window and the cochlea. And you're not getting that amplification if there's a lot of fluid in there. All right? Any questions about the ear? Let me. Okay. I think, let me just, oh no, one more thing. Sorry, this will take about 2 seconds though. And that is talking about the sensors. Of course, we talk about the senses, we know the senses, right? And all of those we've pretty much discussed, but when we talk about touch, we've already talked about the different sensors that we find in the skin. We know about that, and we know now that sensors are what's going to carry that, our sensory nerves are going to carry the message up to the brain. But now, if you move to I'm on page 156. We have some different types of sensors for smell and taste. And this is the part you really miss, because in lab we would get to have a lifesaver, but you could do this at home if you have a lifesaver or some hard candy that you can suck on. If you plug your nose, do you get a good sense of taste? If you have a cold and you're all stuffed up, food doesn't taste the same, right? Well, the sense of taste and the sense of smell are both dependent on chemical receptors. So in the skin, we've had pressure, we've had rods and cones in the eye, we have hair cells in the cochlea. All of those account for different types of senses. But actual chemicals bind with either our taste buds, because our taste buds are our taste receptors in our mouth, or we have olfactory receptors in the nose. And both of those are based on chemoreceptors, meaning that they're based on chemicals. So different types of molecules found in water and air when we smell it, for example, but if we drink water, that gives us taste. We have huge amount of ability to taste different things. But our basic tastes are based on sweet, sour, bitter, and what's my last one? Sweet, sour, bitter,
Salty.
and what?
Salty.
Salty, right. So our taste buds only taste those 4 tastes, but in combination with the smell, we get totally different things that we can taste. What am I trying to say? Our variation in taste.
Why would spicy not be one?
That's a really good question. I don't know why spicy isn't one. I don't know if it's a combination, but I would think it has something to do with the fact that it's triggering olfactory maybe, and not necessarily in the taste bud. But now you got me curious. I'll try to remember to look it up and see. Got to write it down though. So taste buds. We'll see if we can learn a little bit more about those. All right. I think that covers what I want to cover today. Do you guys have any questions?
The summary questions are the only thing that's due.
Nope. Glad you asked though. Okay. So for today, summary questions are due. Then there is one other assignment, and I thought long and hard. I wanted you to take a picture of something. Oh, you guys, I loved the pictures from last week. They were so cool. It really was, I don't know, I enjoyed it. Maybe it's because I got to see all of you and the variety. I figured everybody would pick a couple really, I can't tell you, we've got almost everything on, not quite, but we got the trapezius, somebody had this, and I mean, it was just fun. It was a lot of fun. The Achilles tendon was on one. I mean, people were just very imaginative. So anyway, that was a fun one for me, because these are loads of fun to correct. But this one's going to be a little bit harder for you, but it is something that I always ask on a quiz. And that is, you have to choose either the path of light through the eye and all the way to the brain, Okay, so ending with the lobe that it goes to in the brain, or the path of sound, starting coming through the pinna and ending up at the brain, a lobe of the brain. Okay, so you just need to write those down, and you should be able pretty much to look at your model and write those down, and then that's just a submission, a text submission for it. Okay,
You just pick one of those like sentences and write it down?
well, not sentence, well, yes, it can be sentences, it can be this to this to this to this to this, that's fine. Yep,
Oh, you're writing what the path is.
yes, you are.
I understand.
Yep. So you got to label the parts, basically. You don't need to, you don't have, I say label, you need to know the parts in order. Okay. Any questions? All right. Well, you guys, then I will stay as late as anybody wants to, if you want to stay and work on your summary questions or your assignment. Otherwise, you are free to go. And I hope you guys have a great day. I'll see you bright and early tomorrow morning.
Tomorrow.
Bye, Rose.